I'd like to thank the Focused Ultrasound Foundation for the opportunity to present this research, Focused Ultrasound for Nonviral Microbubble-Mediated Gene Transfer. Developing non-invasive non-viral gene transfer technologies that can target specific anatomical regions would be valuable for advancing gene therapies. We hypothesize that a combination of focused ultrasound with microbubbles may enhance transient vascular permeability in target organs, permitting the delivery of a more concentrated dose of a therapeutic gene, which could potentially resolve the limitations of low transfection efficiency associated with non-viral-based approaches. This technique must first be optimized for each target organ, including the liver and the heart for these studies. And then these optimized methods can be applied to evaluate cardiovascular-related gene therapy for treating liver and heart-associated diseases, including hemophilia B, familial hypercholesterolemia, and angiogenic therapies for myocardial ischemia. We refer to this gene delivery method as UTMD, or ultrasound-targeted microbubble destruction, in which bioactive molecules such as negatively charged DNA plasmids encoding a gene of interest are added to the shells of cationic lipid microbubbles, which are filled with a heavyweight octafluoropropane gas to decrease blood solubility. The DNA-bound microbubbles are delivered intravenously to the target organ by acoustic cavitation at a resonant frequency of the bubbles. In previous studies, we used unfocused transducers with UTMD to optimize gene transfer and were able to achieve successful blood coagulation, which approached therapeutic levels in a mouse model of hemophilia B. More recently, we have incorporated FUS transducers into this work to further optimize transfection efficiency and explore other targets. These ultrasound images show microbubbles in the mouse heart in the top panels and in the liver in the bottom panel before and after microbubble delivery. In the bottom video, you can see the arrival of microbubbles in the major hepatic vessels and liver vasculature. At this point in the technique, external ultrasound is applied with an unfocused or focused transducer to mediate cavitation and transfection of hepatocytes with DNA. In recent studies, we incorporated FUS technology into UTMD using a 1.1 megahertz transducer equipped with a modular water coupling cone that allows us to modify the targeting depth of the focal point between 5 and 18 millimeters. These optimization studies involve delivery of liver-specific and CMV-based reporter constructs to the liver and heart in wild-type mice. In vivo and ex vivo bioluminescence optical imaging and immunofluorescence assays were used to evaluate transgene expression, intensity, and localization. The optimization experiments involve modifying various ultrasound parameters, including the amplitude signal, pulse repetition frequency, pulse duration, focal point targeting depth, and overall treatment duration time, with the goal of identifying optimal parameters that maximize transfection efficiency while minimizing UTMD-related bioeffects. We performed extensive optimization studies in vivo in which modifications to the different parameters were evaluated in separate focused UTMD treatment groups after hepatic delivery of a liver-specific reporter plasmid. The in vivo average radiance data from hepatic transfection with the reporter constructs is shown from the most effective treatments. The highest levels of expression 48 hours after focused UTMD were detected in groups 1, 4, and 6 compared to control-treated mice. We identified the parameters from study 6 to be most optimal for the liver, which incorporated a lower pulse repetition frequency of 14 Hertz, short pulse duration less than 20 microseconds, and longer insonation time of 4 minutes, which allows for more exposure of the liver to focused ultrasound as the small focal point is scanned manually across the liver. Study 6 also incorporated targeting focus energy at multiple depths of 5 and 13 millimeters in a single treatment, which likely permits more exposure of hepatocytes to ultrasound. Importantly, these parameters were further selected because they resulted in fewer ultrasound-related bioeffects compared to the other studies. We observed no significant changes in alanine transaminase activity, which can indicate liver damage, in the plasma of these mice compared to normal, untreated plasma when examined 48 hours and 2 weeks after focused UTMD. These immunofluorescence images are liver samples from the optimal treatment group stained in transverse orientations to evaluate the depth of transfection. Robust patches of luciferase expression were observed in clusters of hepatocytes throughout the liver, and the pattern of expression shown by the white arrows in panel C reflect transgene delivery achieved at multiple depths for a single treatment. As a side-by-side comparison, this observed pattern of robust patches of transgene expression from focused UTMD is distinctly different from the more diffuse and uniformly distributed pattern of expression that is typically observed for unfocused UTMD. We then compared the newly optimized focused ultrasound approach to unfocused UTMD in an extended set of liver-directed experiments and observed highly similar levels of hepatic transgene expression 1, 5, and 8 days after treatment. We also replaced the liver-specific reporter with a ubiquitously expressing CMV reporter to further evaluate if transfection of other liver cell types, such as the vascular sinusoidal endothelial cells, in addition to hepatocytes, would occur more efficiently with the FUS-based approach. However, similar to our previous study, we again observed comparable levels of expression for both methods, 1, 5, and 8 days after transfection. We then used a novel nonlinear contrast imaging method to examine microbubble dynamics in the liver in real time to compare cavitation mechanisms for both methods. Our gene therapy transducers were used to direct cavitation, and imaging was performed before and after treatment to assess contrast signal intensity changes. A microbubble perfusion curve graph is shown on the left for different hepatic vessels and liver tissue compared to non-target control regions. Hepatic perfusion of the DNA-bound microbubbles was quantified by evaluating perfusion pre-delivery, immediately after delivery, and again after cavitating microbubbles with UTMD. We observed a robust difference in microbubble signal intensity changes with a much greater difference detected after focused UTMD, indicating less microbubble persistence as depicted in the bottom right-hand image, and a greater extent of microbubble destruction occurring after treatment with FUS. While this significant difference in cavitation efficiency was observed in the area of insonation, both approaches still led to similar levels of overall transfection. We then switched targets and applied UTMD to direct transfer to the heart using the optimized liver parameters, except treatment time was reduced to 90 seconds and only utilized the 5 mm offset for focal point targeting, as deeper targeting would insonate regions below the heart. The FUS transducer was scanned over a much smaller region of the left side of the thoracic cavity to direct transgene delivery to the left ventricular myocardium. We performed unfocused UTMD for comparison and observed some of our most interesting findings. In contrast to our observations in the liver, significant differences were observed in transfection efficiency with substantially stronger average radiance intensities, greater than 10-fold for focused UTMD when assessed 1 and 5 days after treatment. Serial cross-sections spanning the entire heart samples were evaluated by immunofluorescence to characterize the location and depth of reporter transgene expression in the heart. Luciferase expression was predominantly evident in the left ventricle of the myocardium and was again localized to small and highly transfected regions within the heart. H&E staining was performed to assess tissue effects from the FUS exposure. Some red blood cell extravasation was evident in myocardial regions proximal to regions of luciferase expressing cells, which can often be observed in the first 24 to 48 hours after UTMD. To further evaluate the safety of this approach, troponin leak into the plasma as an indicator of cardiomyocyte damage was assessed with Western blotting and revealed a very faint band corresponding to treated plasma from a mouse that also had the strongest cardiac reporter expression. This finding supports previous observations with which stronger transfection is often associated with increased bioeffects, emphasizing the need to meticulously tailor this approach to each target. With further refinement of cardiac targeting parameters and assessment of acoustic bioeffects, this data suggests the potential for using FUS and microbubbles to non-invasively direct cardiac gene transfer. In summary, we observed similar levels of reporter expression in the liver with FUS compared to unfocused UTMD 1, 5, and 8 days after transfection. Optimal hepatic expression was observed with double depth targeting at 5 and 13 millimeters in vivo, and it incorporated a low duty cycle, low pulse repetition frequency, short pulse duration, and longer insonation time. Robust clusters of reporter gene expression were observed in distinct regions of the liver after FUS compared to the more diffuse pattern of expression observed for unfocused treatments. We observed fewer ultrasound-related bioeffects when assessed histologically and serologically, and nonlinear contrast imaging of the liver before and after insonation showed a substantially greater change in signal intensity for focused UTMD, suggesting distinct cavitation mechanisms for both approaches. And for cardiac delivery, 10-fold stronger levels of reporter expression were observed for focused UTMD compared to unfocused delivery. Future directions of this research include further refinement of focused ultrasound optimization for specific target organs, most recently for the heart and the kidney. Importantly, we are currently exploring the application of these optimized methods for preclinical gene therapies, including the delivery of therapeutic blood coagulation factor IX, as well as novel factor IX variants for the treatment of hemophilia B, the delivery of novel base editing constructs for restoring low-density lipoprotein receptor function in models of familial hypercholesterolemia, and the delivery of oxygen-stable hypoxia-inducible factor constructs for angiogenic therapy for myocardial ischemia, as well as for the treatment of the erythropoietin-deficient anemia associated with chronic kidney disease. With that, I'd like to thank the Focused Ultrasound Foundation for the opportunity to present this work. This research was performed at the University of Hawaii Center for Cardiovascular Research, located at the John A. Burns School of Medicine under the supervision of CCR Director and Project PI, Dr. Ralph Showett. I'd also like to thank the following people and funding agencies for support of these studies: